All right, guys, so we're going to go over notes on mitosis. There's a lot of stuff, but it's really not more complicated. There's just a couple extra terms from regular biology that are a little bit different, but other than that, it's really not more complicated. So we're going to try to kind of fly through this a little bit. And with the video, you can pause wherever and kind of take more time. Okay? All right. So the cell cycle, woo! So when you began, you were that little tiny dot, that little glisten in your biological mother's eye. That's who you were. We have to do mitosis in order to do a couple different things. One thing is we have to be able to make new cells. So, like all the dust in your house, whatever, that's just skin cells that have flaked off of your body. You have to regenerate those skin cells. Otherwise, you would just be open wounds all over your body. No good. We need to be able to make new cells for asexual reproduction. So if you are a unicellular organism, this is how they actually make new organisms. So this is the way that like bacteria actually multiply. We have to be able to grow. For the sake of your mother's birth canal, you did not come out the size that you are right now, right? You grew, and we have to be able to repair and renew ourselves. And that looks different for all creatures. So, like above me, this little weird sausage thing coming out, that is actually the leg of a starfish that got cut off, and it is regenerating a brand new starfish. Not all organisms can do this, obviously, but some can, and it's really, really cool. So when we look at the cell cycle, we are looking at the cycle of what the cell does when it's just kind of like existing, and we call that interphase. The actual mitotic process, that's where we get prophase, metaphase, anaphase, telophase, and then cytokinesis. Those are all kind of different stages of that same thing. So just to kind of like review some other stuff, DNA is in your nucleus, and your cytoskeleton becomes really, really important in this process because we make something called microtubule spindle fiber. It's microtubule spindle fibers. These spindles are made out of the same proteins that like the cytoskeleton is made of, and they actually help to pull the chromosomes apart. When we're looking at DNA, and we're going to look more at this in our next unit, so this is kind of like our intro to all of this stuff, which is kind of nice. The nucleus protects our DNA, so in order to actually replicate our cells, we have to break down that nucleus because we have to be able to get that DNA out. When we're talking about like transcription and translation, when we're making proteins from DNA, in that process, we have to have a different molecule that brings the DNA message out to the ribosomes, which is RNA. But here we have to completely degrade that nuclear envelope. So when we're just looking at how everything is set up, typically the nucleus is going to have like those pores, so RNA can come and go, but the DNA cannot get out of that. Little bit of background at the cytoskeleton. This is made of protein fibers called microfilaments or microtubules, intermediate filaments. They're all just different protein arrangements of very similar structures that all act as part of the cytoskeleton. Parts of the cytoskeleton are responsible for cell movement, so the cilia, flagella, and they're also responsible for regulation. So they're going to actually be like the highway to all of the different parts of the cell. When we're looking at this picture of the cytoskeleton, it makes a little bit more sense. So in this picture right up here, we have, for instance, like a mitochondria that's hanging out, and we can see all of these little intermediate filaments that kind of hold everything in place. A lot of times molecules will actually walk along these filaments and these microtubules to actually get from place to place inside of our cells. How that works is actually really, really cool. And it works with ATP, of course, as everything does. So if you have a tubule or a filament like this, and we have some sort of a walking protein, it's going to attach. So it's going to make a new bond. It's going to physically swing the back end around, attach this part. So it kind of like, or maybe I should do it with my fingers. So it's like this attaches, this attaches, this attaches, this attaches. It's actually little things that walk along. It's very weird, very cool. But this is how we actually transport things within our cells, and this is how we transport vesicles. So we don't typically think of, or we typically think of vesicles as like a little bubble. It just goes like and through your cell. It actually walks along these filaments. Here's where this becomes important in cell division, is these little structures called centrioles. These centrioles are going to be in animal cells, and they are a little bundle of microtubules. I think they look like little licorice, kind of, sort of. And these little guys with spindle fibers that come out of them, all these things are made out of protein. They are actually going to physically guide and manipulate our chromosomes, as you see in that picture on the side, in order to separate them in that process of mitosis. In order to get all of the right stuff, this is like a highly, highly choreographed dance. We need to be able to get an exact copy because we make two identical daughter cells and sister cells. So the process of getting the genetic material split is called mitosis. This is an important part, so like star this in your notes. The division of organelles in cytoplasm is called cytokinesis, and it's actually a separate process from mitosis. Overview of this thing. You guys should know this, or it should look very, very similar, and it should only take a little bit of a refresher. So we have interphase, that's when the cell is just chilling. Prophase, we can start to see chromosomes condense. We can start to see those spindle fibers, so those extra little protein pieces are extending. Prometaphase, it's one of those things, some people include it, some people don't. It's like the intermediate, but there's always going to be an intermediate phase because stuff is like moving in between. Metaphase, they're all lined up in the middle. When I say they, I mean chromosome pairs. Anaphase, they split. And cytokinesis, we are physically splitting the organelles and the cytoplasm. It's kind of like telophase, cytokinesis happen at the same time, because with telophase, we're splitting fully the nucleus. Cytokinesis, we're fully splitting that cytoplasm piece. Most of a cell's life is spent in interphase. 90% of a cell's life, in fact. This is just when they're vibing, as the kids say, I hear on the TikToks and whatnot. So they just vibing, they just hanging out, having a good time, and then they're gonna hit certain checkpoints of growth, of enzyme regulation that is going to trigger them to want to divide. Okay. So with the cell cycle, every life has some sort of a cycle to it, right? With this, we are either doing one of two things. So when a cell is formed, it either grows and matures to divide again. So it keeps going through that process of growing, dividing, growing, dividing, growing, dividing. These are things like epithelial cells. So your skin cells are constantly dividing. Your blood cells are constantly dividing. Fun fact, one of the reasons why your poop is brown is because most of your poop is actually dead red blood cells that your body gets rid of, and the oxidized form of that iron in your fecal matter looks kind of brown, like rust. Yeah, that's one of the reasons why your poop is brown. There's your fun fact for the day. Share that one at the Thanksgiving dinner table. So when your cell grows and matures and never divides again, there are some cells that will do that. They'll just keep growing. Your nerve cells, your muscle cells, they don't necessarily never divide again. That's kind of a misnomer. It just takes them a very, very long time. There's some evidence that some nerve cells can take up to 24 years to get the memo to probably divide. This is the reason why spinal cord injuries and brain injuries are so detrimental, but a laceration in your arm isn't, right? Because of different times of repair for these different cells. Liver cells are kind of in the middle. They are very, very unique in that your liver cells just kind of like chill and hang out, like, look at me, I'm a liver. However, if you get like a laceration on your liver from a traumatic accident, or if you're doing a little too much of you know what I mean? Your liver, when it is not in those conditions anymore, can actually repair itself. It's one of the few organs that can actually regenerate. And that's one of the reasons why liver transplants, you take like one lobe of a liver and put it into somebody, and they can grow a whole new liver from it. The liver cells can kind of do a lot. And these are kind of some recent developments in that. Kind of cool. Okay. Deep breath. So interphase. Remember, 90% of a cell's life is spent in interphase. We have it broken down into 3 separate parts, and each part has some sort of a checkpoint before it can go on to the next part. So our first one is the G1, or the gap phase. So this is just where cell is hanging out, it's growing. It's going to get to some part of growth where all of a sudden it's like, maybe I'm too big for one cell. Maybe I should be 2 cells. What happens is it's going to send a signal to the DNA to start replicating itself. So it's going to be like, hey, we should probably like make some copies of this DNA business. Your DNA is going to make a copy. It's going to copy all the chromosomes. That's our S phase. That's our synthesis phase. G2 is when we're preparing for division. So the cell is going to grow even more. It's going to be making extra organelles, making extra proteins, making extra membrane that's going to put some vesicles. It's making all that extra stuff. The little G0 is off to the side here because when a cell is just existing and not getting ready to divide, like those nerve cells or whatever, we call that G0. So that's when a cell isn't really getting ready to divide. It's not really growing. It's just kind of hanging out. If we look inside into that nucleus, as we're preparing for mitosis, we're going to be replicating all of the DNA and the proteins in there and produce more proteins and organelles. In your nucleus, all of your DNA is in really, really loosely packed molecule called chromatin. We're going to look more at how that all works. So when we are copying our DNA, and we're going to go more in depth in this in our next unit, this is the synthesis phase of interphase. We are going to replicate our DNA. So if we look at the picture up on the top, we have our double helix. We take each side of that double helix and add new bases onto the other side in order to fully replicate that molecule. So when a human cell duplicates, every daughter cell gets an identical copy. Yes, we do have errors. This is where mutations come from. However, most of those errors are going to happen in non-coding parts of our DNA. There's a lot of junk, quote unquote, in our DNA. So most of our errors happen then. And some scientists think that we have so much junk in our DNA to thwart against these errors actually happening in a gene that we need. When we're organizing our DNA into chromosomes, we have several different levels of organization. So we have these little proteins up here called histones. These are going to be super, super important when we talk about some really cool genetics stuff. These histones have DNA wrapped around them, and then they're coiled together. So think of a histone. So like a little spool of thread here. So imagine this thread, you can kind of see a little bit, is like our DNA, and it's wrapped around my little spool. This is in order to keep it organized, because you can imagine in my little sewing kit that I have here, how messy this would be if I just had thread and no spools. Oh my goodness, I would go into crazy land. But with all those spools, we keep everything nice and organized. From there, into those organized bundles, we get chromatin. From there, we coil, and they call it supercoil when it's like a coil within a coil. So it looks like those old school telephone cords. You guys probably have no idea what they are, but whatever. Then that is what actually forms chromosome, as you have seen them in pictures. So the chromosome that you see is about half DNA and half protein all wound around. So this is more pictures of that process going in that condensed chromosome. So when that DNA replicates, one side of this chromosome is exactly identical to the other side. So this, when they're connected together, is still called one chromosome. When we rip them apart, it is now 2 chromosomes that are identical. So these are double-stranded mitotic human chromosomes. Each side, when they're connected, we call a sister chromatid. Okay, sister chromatid. They are connected at the centromere in the middle. That is the centromere. and they have identical copies of the new DNA. When we're getting into the actual process of mitosis, we're dividing a cell's DNA between two daughters. And a lot of people call this a dance of the chromosomes because it is very, very regulated. So let's go through the steps. So in the first step, which is prophase, we can actually start to see visible chromosomes or chromatids. The centrioles, remember those microtubules, those things are going to move to the opposite sides of the cell and start to send out their spindle fibers. What happens then is the nucleus is going to start to break down, and then those spindle fibers are eventually going to connect to each of those chromosomes to coordinate their movement. Okay, so really, really super important, really cool stuff. In the transition to metaphase, so some people call this prometaphase, the spindle fibers will attach to the centromeres and form something called a kinetochore. Kinetic means movement. So this is how they actually physically move. So down here, this little central part with the spindles, that is the kinetochore. Here is how the chromosomes begin moving so they can all line up in the center. In metaphase, chromosomes are going to line up in the center. Metaphase plate, meta means middle. All of these different things are going to happen when they're in the middle. Then we can ensure that our chromosomes are going to separate properly, because in each new nucleus, we need to get half of each chromosome. So everything lines up in the middle. We have like our nice line across where they all go. Each side is a sister chromatid of the same chromosome. Once they separate, then it becomes two new chromosomes. Anaphase, we pull and we push. So if you look at the picture here, you see some of these spindle fibers are attached to each other. Those spindle fibers are pushing against each other, while these other ones are pulling apart. So it's a push-pull to actually get these apart from each other. And they're pulled together by motor proteins that kind of walk along, just like how muscle cells contract, if you ever look at that. And this takes a ton of ATP to be able to do. As those poles move apart, so our cell is starting to kind of elongate. When we separate these things, this is the part where people kind of get confused terminology-wise. So on the bottom here, we have one chromosome separated into two chromatids. Once they get separated at those centromeres, then it is two individual chromosomes. With that chromosome movement, this is kind of how it's set up a little bit. So the kinetochores will actually physically walk the chromosome where they need to go, and that microtubule is going to shorten. I like to think of it as like a telescoping, like fishing rod, if you've ever seen one of those, or like a antenna in some like really old cars is like telescoping. That's kind of how it like walks itself back to where it needs to go. In telophase, we finally get two new daughter nuclei that start to form. The chromosomes kind of start like relaxing a little bit. They're like, oh my gosh, all that's done. I did my dance. So now they all start to kind of separate. And then cytokinesis starts. So remember, cytokinesis is the actual separation of the organelles and the cytoplasm. So we start to get what's called a cleavage furrow, because to cleave means to separate, like a meat cleaver, karate chop, right? It's going to separate, or cleavage, right? Like a butt crack. So all of those different things are going to separate, and it pinches in like a little string in the middle, and it actually separates our cell into two. You can see here up above is a electron microscope image of a cleavage furrow in an animal cell. So all of the little like parts of the cytoskeleton inside of our cell help to like pull together those different parts of the membrane. Very, very crazy cool thing that happens. There's tons of videos of this happening on YouTube. I'll try to remember to link some in the Schoology for today. So this is the whole process when it's actually physically happening in a cell. So interphase, Prophase, notice how we can start to see those condensed chromosomes. Metaphase, we clearly have that line of chromosomes in the middle. Anaphase, they're beginning to pull apart, so anaphase is kind of like a mix between these two. And then late telophase and cytokinesis is when they are finally separated from each other. Once again, just some more pictures of that happening. Just with, this is different microscopy, so this is fluorescence microscopy. In plants, it's a little different how this happens because if, as you can imagine with a cell wall, we can't like pinch in a cell wall. So what happens is the Golgi apparatus will actually package cell wall materials. And what happens is those vesicles will then go bloop, 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 and they line up in the middle after those chromosomes are separated. And then all of those vesicles kind of release all of their cell wall materials and they make a cell plate in the middle. So we have that new cell wall that's laid down between the cells after that inner membrane pinches off, and that's how we create two new cells. So a cell plate is formed in plants because they need to make that physical wall between the two cells. So here is a picture of that happening. So we can see all of these little like bubbles that line up. Those are all vesicles that have cell wall materials that are lined up in the middle. This is just mitosis happening in plant cells. So just kind of what all of those things look like under some different microscopy conditions. And here's another one. So this is what it looks like in an onion root hip. So as we can see, like these cells are just an interphase. They're just hanging out. I would say this one and this one are probably in prophase, metaphase right here. Up here we have some anaphase happening. I don't see, maybe this one I could say would be in telophase because we see one cell wall around here and it's probably in the process of laying down that plate in the middle. They can kind of go either way with some of these, but this is kind of what all those things look like. So with mitosis, we think that this evolved from binary fission in bacteria. So bacteria mitosis is called binary fission. It happens almost exactly the same, except for bacteria just have one circular DNA. So they just have to replicate that one circular DNA and then separate that one little DNA and some of their extra plasmids. But it happens almost exactly the same. And it's a little bit faster because they have no membrane-bound organelles to actually replicate as well. Because a lot of their life processes happen in their cytosol. So with this as well, there are some protists, so like little single-celled organisms, think like amoebas and things like that do have some microtubules that help to start them actually separating their chromosomes. So this is kind of a progression of how we think mitosis evolved in animals up to higher species that we see today. So that is it. Ooh, flew through that. Good job, guys, sticking with that. And I will see you later.